So hello and good morning. It's time to start talking about the digestive system. And we talk about respiration and digestion together because they are always constantly linked. The digestive system is the system that acquires and processes our fuel in the form of carbon molecules. Everything that we eat, except for salt, is a living cellular tissue. And its nutrient value rests in those polymers that make up the bodies of our food. Now, a contrast that I want to point out right away is that whereas the respiratory system structurally is like a cul-de-sac, you turn into the cul-de-sac, you drive to the end, you turn around and you come back out the same way. The digestive system is a thoroughfare with an entry end and an exit end. And under normal circumstances, healthy circumstances, it's one way. What I'm going to emphasize is that we eat these nutrient materials, what we call food, in bulk quantities. And in order to digest it and use it as food, we have to break it down. Now that includes a combination of things. I think the first thing I would mention, especially for human beings, is the social and cultural practices that we have surrounding our food. First of all, we select food and we breed food. We develop food materials to be the purest form possible, high in nutrients, high in carbon-carbon bonds, so that when we break them down, we get lots and lots of energy out. The chemical bonds in our food is going to be captured in our aerobic respiration system and transferred to ADP. When we phosphorylate ADP, we make ATP, and then we have the material to support our metabolism. So we're going to break it down. We're going to first of all select our food items. That external business is food science. But then we have on-board material, notably our teeth, that begin mechanical digestion, and then the muscular contractions that mix and churn those progressively smaller and smaller bits, add to the mechanical breakdown of our food. And what we're doing there is increasing the surface area. A block of food has a certain amount of surface area. Cut that in two and it has more. Cut those in two, it has more. As we get to smaller and smaller particles, the surface area goes way up. And the reason we're doing that is to expose molecules throughout that initial block of food to chemical processing, and that's the second step. There are molecules that carry lots of energy, such as, for example, the polymer starch, which is abundant in plant food materials. In fact, a lot of the plant materials we select, we use because of its high starch content. But starch is a polymer. Starch cannot be digested by cells and used in aerobic respiration. It has to be broken down to its monomer glucose. And so we have chemical enzymes that are specific to the breakdown of the various polymers we've named. So back when we talked about carbohydrates, and we talked about starch and cellulose and sucrose, all of those things have glucose content, but we know cellulose, because of its bonding, is indigestible. But starch is loaded with readily obtained chemical energy. The same thing applies to sucrose, by the way, our table sugar, which we eat in great quantities and we actually seek out because of the taste. But it's a dimer, actually. It's glucose and fructose. We have to have an enzyme called sucrase to break it down into monomers, and that's what we will digest. And that's the second part. Mechanical digestion is followed by chemical digestion to actually yield the monomers that will be nutrients within our body. So if you think about a food label on the back of your power bar, that food label contains carbohydrates. It often separately lists sugars, and those are in various forms. So within that number that's registered, it will contain the total content of things like starch, things like maltose, things like sucrose, and it is the monomer content, the monosaccharides, that our cells will use. Likewise, when we talk about a protein content in our food, it isn't the protein that we will digest or use to synthesize our own proteins. It is the amino acids. And amino acids require some additional processing. Amino acids release ammonia when they're processed, and so we have an additional metabolic cost to digesting proteins. Finally, we have fats, that hydrophobic component that is basically chemically very, very reluctant to mix with water. We're going to use that in many ways. One of the first ways is as phospholipid, where the job is to aggregate into sheets and form barriers like our membranes. But a second product of eating fat is that we will take those fatty acid chains and hook them to a glycerol and produce a triglyceride, our own transport form, which will sustain aerobic respiration by providing acetyl groups. We're going to get that by a process called beta oxidation of the fatty acid chain. So that overview kind of all surrounds the fact that human beings, like all animals, are heterotrophs. We are other feeders. You can put us out in the environment, and the sun gives us a sunburn. The CO2 in the air is just an inert portion of the air we breathe, whereas a plant's taking water and carbon dioxide in that sunlight and actually making carbon monomers and carbon polymers as it builds its body. So this will kind of give you an idea of the system that is meant to find, identify, and acquire food, put that food into our digestive tract and process it, and free the nutrients in monomer form. Now, from that point, the big issue is absorption. We're going to absorb those monomers. We're going to take them into our body and transport them all over the body, process them for ATP production, yielding the energy that we need to run our metabolism. But also, the monomers are the building blocks of our own proteins, of our own fats, and of our carbohydrate structures. So it's a fact we are other feeders. We are heterotrophs that tailors the digestive system. So what are we going to do? We're going to break down food into nutrient molecules that can be absorbed across a membrane. We're actually extracting them from the digestive tract. Now, the digestive tract is not inside the body. It is surrounded by the tissues, but if something's in the digestive tract, it's actually just moving through a hole in the body. And you have to cross a membrane before you enter the tissues. We're going to deliver these nutrients to the cardiovascular system, and I should add something, to the lymph system. It turns out that transport of carbohydrates and transport of proteins or amino acids occurs by direct delivery to the blood. But fat, oils, waxes in our diet are absorbed not by the capillaries, but by the lymphatic vessels, the lacteals. And it is the lymph system that transports it from the digestive tract walls all the way up to the top of the thorax. And that's where those fat components enter into our blood flow. Now, we know that the food contains then two components, the digestible component and the indigestible component. So basically, as we extract the nutrients and the cofactors and the ions that we need for our metabolism, we leave some things behind. The primary thing we leave behind is cellulose. And cellulose is indigestible, even though it's made of pure glucose. And it moves through the tract, cellulose providing the bulk, the actual structure for our feces. Now, in addition, as this material flows along, the body takes advantage of that flow all the way along the tract to deposit certain waste or breakdown materials. And those move into the lumen and are carried by this waste material to the other end for elimination. Now notice this term. The waste products that leave the digestive tract, we don't say they're excreted, we say they are eliminated. And that relates to this thoroughfare, this hole through the body. They never actually enter the tissue. They basically move through that hole and leave out the exit point. Contrast that with urine, where the ammonia and urea that's produced by digesting proteins is actually circulating around between the cells, moving through the blood, processed to urea in the liver, and finally processed out of the blood flow in the kidneys. It's actually coming from the internal part of the body out to collection in a bladder, and eventually its elimination is achieved by passing urine. Now that's an excretion, but digestive system, we talk about it's an elimination. So we have any number of diagrams that can show you this. Here's the cul-de-sac we call the respiratory system. Down and back. I'd make this point. In the air sacs, the oxygen in the air sacs of the respiratory system was not actually within the body. When it crosses a membrane and enters the cardiovascular circulatory system, then it's inside the body. Here is also where carbon dioxide crosses out of the body and back into the respiratory system. But the digestive system is a thoroughfare, in one end, out the other, interacting with these other systems along the way. Naming the parts of the digestive tract is a very sequential process. It's a list in order. Up here, the mouth is the first cavity, the entry protected by the sphincter muscle and the lips. Basically, an area that has multiple functions, the respiratory system, speech, and breathing all occur in the mouth, as well as digestive activity. But within this cavity, it is the teeth and the tongues that are primary digestive organs. The tongue is a very large and strong muscle. It's adept. It can move quickly to articulate our sound into speech, but it is equally skillful at maneuvering our food so that our chewing is efficient at breaking it down mechanically. So the number of times I bite myself on the tongue is relatively limited to the number of times that I eat a meal. So from this cavity, we're going to enter this back pharynx, the pharynx being the common opening at the back of the nasal, oral, and laryngeal cavities. Here is where the epiglottis manages the open airway and basically closes it at the instant of swallowing. So let me make a contrast. We breathe almost every second of the day. When we swallow, our airway is closed for less than a second as the food moves past the glottis and its opening to the airway and passes down into the esophagus. The next tube is just a connecting tube, and once you initiate that swallow up here, then autonomic control takes over. It is actually involuntary peristalsis that moves the food directly to the stomach very rapidly. The stomach is a bag for the receipt of something from the outside of the body. And so the acid in the stomach is going to first largely sterilize the food, not completely, but largely remove living pathogens, kill cells, denature viral DNA, all of those things that we would like to achieve before the food passes on to a place where the nutrients enter the body. Below the stomach, we have small intestines. Small tubes that wiggle back and forth have much more surface area than large tubes. The decrease in diameter is in direct relationship to that surface area, and it's the small intestine where most of the absorption of our nutrients, cofactors, vitamins, ions, other things is going to occur. But we have crudely processing food entering the bottom of the tract here and entering the large intestine. Now, oddly enough, we're going to climb vertically up through almost half of the abdomen, traverse over from the right to left side, and descend right back down on the left side of the body. Then there's a kind of a wiggle. So we have the ascending, transverse, and descending arm of the large intestines, often associated with the term colon. The sigmoid colon will move the tube toward the midline and posteriorly. So it will snake backwards to position over the rectum, which basically is posterior and situated directly above the opening, the anus, which allows the passage of the elimination from the body. We have a number of organs and actually a number of tissues that are in or around the organs of that digestive pathway. And they have a major digestive function. Because they have other functions, we don't call them primary to the digestive system. We call them accessory organs. Saliva is a good example of the many glandular functions of the digestive tract. Saliva is a protective and lubricating fluid. It has its own digestive enzymes, so we're going to start some chemical digestion right up here in the mouth. Oddly, not oddly, we eat so much starch. Starch is pure glucose. Amylase comes from the glands of the mouth, and we start that chemical breakdown right here. The liver is a large processing organ. Sometimes I think that the hepatic system should be an organ system of its own because of its many important roles. For digestion, it produces a substance called bile. Bile is critical to our digestion because of the hydrophobic nature of fats, oils, and waxes. Bile is what we call an emulsifying agent. It causes fats to break into smaller and smaller and smaller droplets and remain in that broken down form, suspended. That increases the surface area and lets the enzymes that can process fats attack them. So we're also going to produce bile in the liver. By the way, we're going to transport the bile to this gallbladder, and we're going to basically wait for food to come down the pipe, and then we're going to squirt as long as food is moving through. Storage and concentration of the bile in addition to the digestive stream is what the gallbladder does. And I want to step back here to the liver. We're going to use the liver to store nutrients in our daily feeding cycle. So when we eat a big meal at lunch today, there's going to be a time where we're actually absorbing nutrients into the bloodstream, the absorptive stage. But that's over fairly quickly, 45 minutes to an hour and 15 minutes. Most of the usable nutrients of that meal have been absorbed, and now we have 3.5 or 4 hours until our next meal. So what the body does is that during the absorptive stage, the high stage, it stores glucose in the liver. It also is topping off tanks in the muscles if necessary, but it's storing nutrients during the absorptive stage in the liver. And then as the absorptive stage ends, it will break that glycogen down and feed it back into the bloodstream to help regulate blood sugar in the post-absorptive stage until our next meal. The final accessory organ here is the pancreas, sort of the star of our generation, because of the ability to now treat diabetes and keep diabetics alive and healthy. This is an exocrine gland. It secretes many digestive enzymes and hormones that are important in digestion, but the star of the era is insulin. The inability to produce insulin used to be a death sentence. It was type 1 insulin-dependent diabetes, used to be called childhood diabetes or juvenile diabetes. And it was a wasting disease where a healthy 9-year-old, 10-year-old, 11-year-old would all of a sudden just start to lose weight and literally would waste away. This was despite studying patients in all conditions, including patients that had unlimited medical care and unlimited nutrition, unlimited hygiene, and yet they would waste away and die. And it turns out insulin is a single factor needed for the processing of nutrients into the cells. So insulin and its provision now allows us to predict a long, healthy life for diabetes under proper management. And it is the pancreas that produces that insulin. Now let's think about the activities. What's actually going on in the digestive tract? First of all, we do drink liquids. Water is corrosive as it flows, but not so corrosive as the solid materials of our food. The most abrasive stuff that we put in our body comes into the mouth and is processed through the digestive tract. So solids and liquids are involved in the ingestion. After we ingest and do a few voluntary activities, here in the top end of the digestive tract, we're going to position the food, we're going to chew it, mix it with initial enzymes and saliva to lubricate it, position it at the top of the pharynx to the middle part of the pharynx where we can hold it voluntarily until we initiate a swallow. So we're propelling it voluntarily up here, but when we initiate that swallow, autonomic conditions take over and start moving it through the tract. And that's going to be the case, autonomic movement or propulsion all the way through the digestive tract until the elimination, the feces are deposited into the rectum. Now there is a signal when the rectum gets full and swollen. Stretch receptors say it's time to eliminate, and there is a voluntary element to that elimination. And mostly, though, to the release of the sphincter and the initiation of that elimination or defecation. So propulsion is a second activity. Along the way, there's progressive and continuous. Mechanical digestion, that's the physical breakdown of the food into nutrients. And chemical digestion, this is the specific enzymatic breakdown. And so we see how specific this is when we think about a particular kind of food. So carbohydrate, polymers such as sugars and starch are broken down progressively from polymers, meaning unlimited length of the chain, to saccharides and disaccharides, and then eventually to monosaccharides that can be digested. Now, lactose intolerance is something that is related to lactose, the milk sugar, which is a dimer. You have an enzyme called lactase that breaks it down into two monomers, and you can digest those monomers. If you don't have the adult form, it stays as the dimer. It continues to pass with the digestive material and produces a range of difficulties in your digestive tract, anything from mild gas or mild indigestion all the way up into vomiting or diarrhea. So lactose intolerance is related to this chemical digestion step. Likewise, we have an enzyme called maltase, which breaks down what's called malt sugar, maltose, and it's a dimer that is broken down. I've already mentioned sucrose. It's broken down by sucrose. Once this digestion is completed, absorption of transportable nutrients. Sometimes you can absorb a disaccharide even though you're going to break it down to monosaccharides to use it in the cells. That absorption occurs principally in the small intestine, although it does trail off. Late in the small intestine and in the large intestine, we are able to extract material from the digestive flow. We are able to reclaim water in the lower part of the digestive tract, and our digestion in the lower part of the tract is also aided by an abundant population of microorganisms, our so-called gut flora. Down here at the end, we accumulate the material that's left behind, the indigestible material, and eventually eliminate it by defecation. Now, in order to control and manage this digestive tract, we have here I think this is supposed to be a mid-sagittal section through the body showing the principal part of the digestive tract. We're going to have a tube that varies in diameter. The esophagus is narrow, the stomach that you see here high inside the abdominal cavity. This is the diaphragm. This stomach is suspended high by mesenteries, double sheets of membrane, a serous membrane system, and we call this the peritoneal membrane. And like other double membrane systems, we're going to have a layer that basically coats or lines the outside of the organ. Here you can see liver, and it is basically covered all over by this outside red visceral peritoneum. It then gets to the double fold that you see here, producing a sheet. Now you would see this sheet as a kind of a webbing sheet that stretches from the liver down to the stomach. This provides support and physically holds the stomach in position. Now, it's a flexible membrane, sort of like saran wrap. It can stretch some, and so it provides for flexible strength and support. You get to the stomach, and you see the visceral peritoneum surrounds that stomach and forms another double sheet, sort of like an apron that falls down here, basically comes back and starts. This is probably the transverse colon, or the transverse large intestine. Here is the mesentery again to an attachment point. And it needs that attachment point because it's going to suspend weight below. Notice that it moves along and forms this rim that basically is attached strongly to the inner wall of the abdominal and pelvic cavity, shown in blue. And that is the parietal peritoneum, a support structure. Notice that there is a space behind the peritoneum. This is called the retroperitoneal, or behind the peritoneum space. And you can see here the pancreas is outside the peritoneum. Also, this is the duodenum of the small intestine. It loops out after the stomach and then comes right back into snake back and forth in this lower abdomen and form these lower small intestine portions. Now, this particular section has kind of shown you the way the serous membrane, the peritoneum, doubles, and then when it gets to a portion of the small intestine, it forms that visceral peritoneum around each tube, each section of the tube all the way down. So this kind of holds and suspends the organs of the digestive tract. Now, think about the digestive tract. It's a tube. To work, it has to stay open. The muscles have to work. If you took away that support of the mesenteries, it basically would fall into a pile, and the heavy portion would press down and constrict or cut off food flow. So as we've seen before, the actual placement is determined by this peritoneal cavity. We see another section that shows you the continuation. Notice that the rectum here and the uterus in this female section, the urinary bladder in both sexes, are both below the peritoneum, and the suspension of the small intestine here is the sigmoid colon coming over to the rectum. The small intestine, the transverse colon, you'll notice in this anterior position, and the stomach above it, the liver located above that, right below the diaphragm. Here is the pancreas and the duodenum, the first, let's just say the first 6-7 inches of the small intestine, which has looped out into the retroperitoneal space, then enters right back in to join with the lower small intestine. When we look at the wall of the digestive tract, we see a very sequential and very logical ordering of the membrane, starting with the serous membrane, what we call the serosa. We see outside a connective tissue layer and the peritoneum itself. Now here you can see this mesentery in its sheet-like form. See how when it's wrapping this portion of the tube, it also provides not just this flexible sheet that suspends the whole tube, keeps it from dropping into a pile, but also provides structural supports for the tube-like structures of the blood vessels, arteries, and veins, and the nerves shown here in yellow, because yellow nerves have to penetrate several layers of this digestive tract. So you'll notice how they come here between the muscles, two muscular layers that form the muscularis, and they fan out to provide the neuromuscular junction that can stimulate autonomic contraction and relaxation as we produce these muscular waves called peristalsis. Now, when you have two layers like this, the layers are defined by the way the smooth muscle cells are laid down. So the outer one is called longitudinal because showing the pointer running in a longitudinal way, they run along the length of the tube. So when the longitudinal muscle contracts, the tube gets shorter. Inside is a circular layer so that the muscles are laid down as a circumference at that position. So the circular muscle layer, when you contract that, the tube decreases in diameter and it pushes the non-contracted material on either side away from it. So it gets thinner and longer when the circular muscle contracts. Now, these nerves, blood vessels, are going to permeate this entire structure. Even though they're not drawn in at this magnification, they are providing supplies and nerve services to all living cells in this system. You'll notice that the nerve extends beyond the muscular layer to this next submucosa. Now, mucosa is going to refer to mucus production, and since the mucus production is actually occurring in this inner part, we're going to see this called the mucosa surrounded by the submucosa, shown in blue. You'll notice there is a nerve net or a reticulum that basically spans between the circular muscle and the submucosa. We can see the presence of glands in the submucosa that are producing the secretions that are added at a particular part of the digestive tract. You can bet that there's plenty of mucus all the way through. Finally, this mucosa, showing you the ducts here from a gland in the submucosa, contains these green spots. These are lymph nodules that are placed in the digestive tract, the so-called MALT, or mucus-associated lymphoid tissue. Now let's make that connection to that name, MALT. Every tract in the body that opens to the outside has mucus glands. We call them goblet cells. And that is because we have to moisten and line that tract. Things may be moving in and out. They may be moving, like in the respiratory tract, the mucus escalator brings that mucus and dirt back up to where we can put it in the digestive tract and move it along. The digestive tract moves through. Urine moves through. There's a lubrication that's necessary for reproductive function as well. And so this mucosa is equipped with nodules that allow for the aggregation of lymphatic cells and other leukocytes. Anytime you have a tract that is moving material from the outside to the inside, and the digestive tract is going to have a major portion where there's absorption, there's the danger of contamination or infection. So the presence of MALT, mucus-associated lymphoid tissue, is logical here in the digestive tract. We also have another abbreviation that goes right with that called SALT, skin-associated lymphoid tissue, and the connection is obvious. The skin faces the outside. And so the chance of infection or contamination is reduced by the dry and impermeable nature of the skin, but some things still do get through. And so having a lymphoid tissue distributed in the skin as an element of the skin, again, it makes sense. MALT is shown here, mucus-associated lymphoid tissue. Entry into the GI tract. Let's start the trip. In the GI tract, in the oral cavity or buccal cavity, this is where we have our voluntary, our perceptible special sense, and that is taste. Taste helps us a great deal because we can taste things like materials that are of a basic pH. Bases are very deadly to cells. And when we put them in our mouth, they're very bitter and we spit them out. We don't swallow them. We also can detect the putrefaction that occurs when food substances break down. They become spoiled. They produce acid. They taste sour. They taste just unpleasant because of the combination of chemicals. So taste sensation is a part of this cavity. Mechanical digestion is what we're going to do in spades. Now, we've already selected food substances. We prepare those food substances. We soften them. We precipitate protein through chemical treatments. So, for example, think about what pickling is. Think about when we make jelly or we preserve things in sugar. What we're doing is processing the food to be more digestible and often more appealing. We also sit down at the table after the kitchen tools, cooking, and the knives, the ladles have basically chopped our food into small bits for us prior to putting it in our mouth. But when we put it in our mouth, we're basically going to position it, and we're going to cut it with our teeth. The teeth are a voluntary activity. We can chew our food well, as our mothers suggested. And during that process, mucus and saliva is being mixed with the food. Also, the glands in the mouth produce salivary amylase. Ama refers to starch, and lase means to break down. So we're going to start. Starch is very accessible and very abundant. Flour is largely starch. Sugar is largely starch. And so these digestions are released by the richness of our diet and starting the digestion in the mouth, breaking it down to glucose is logical. We also, though, we're putting material in our mouth, and as thoroughly as we cook it, as carefully as we select it, just carrying it from the stove to the table is going to get some potential pathogens, some debris on it. So in the mouth, we're going to see the addition of lysozyme, an antimicrobial agent, and antibodies that are present to begin the cleaning up of our food. Now, mentioning again a reminder, respiration and speech are also important in the oral cavity. But normally when we're chewing, we don't use our airflow. We can breathe while we chew by using the nasal cavity, but we do that largely by shutting the oral cavity off. And don't chew, don't speak with your mouth full is a good indication. It can lead to choking, but it also garbles your speech. This cavity is one that is the mandible and the maxilla provide the mounting depressions for the individual teeth. They are surrounded by soft tissue, the tongue in the base, and a kind of a bilateral division as we see these tissue sheets called the frenulum, the labial frenulum here at the lip, joining at the midline, the lingual frenulum below the tongue, and opening to the ducts of the glands that provide saliva and mucus and other substances in the mouth. This midline section, mid-sagittal section, is meant to give you a different view of the separation of this nasal cavity. We talked about the hard palate. Here's the palatine bone back here and the palatine process of the maxilla here. The teeth shown only in the incisor region in cross-section, but the surrounding of the other teeth and back to the molars shown on the right side. The body of the tongue is extensive in its basic connection here to the inner portion of the mandible, and the positioning of the hyoid bone, showing you, I think, in the best way possible, how the hyoid interacts especially with this larynx. See how it is tied to the epiglottis? And then it forms a U, so it would come out toward the pointer and curve back in this direction to surround, in this case, the left side of the trachea. But the main thing about this that's useful is the positioning of these tonsils and salivary glands that are shown, and the joining of the pharynx, the nasopharynx, which is normally open for breathing. The airway is open almost all the time. And this flap right here called the uvula, that part that you see sticking down when you look in the back of someone's throat, is a mobile flap that can actually move up and separate the nasopharynx from the oropharynx below. This is important when you swallow. When you're pushing that food back, if you just contract muscles and this is open, it would squirt that food right back up into your nasal cavity. But the uvula can seal that off as you're positioning the food here in the oropharynx. Now, at that positioning, even through positioning and holding, you can breathe around that. But when you move this food down, there's the danger that it will enter the airway. This is where the epiglottis is going to flop down, and the esophagus and the oropharynx are actually going to bulge. It's going to basically move over this like a roof and drop off the eave on the other side as the food enters the esophagus. We do see tonsils, which are fancy kind of lymph nodes, and their size and their susceptibility to infection, their response to infection, are well known in children especially. Before there's a lot of immune cells, we do have the presence of these tonsils, and tonsillitis is a common childhood disease. In my youth, tonsils were simply removed after the third or fourth case of tonsillitis. Now the medical tendency is to retain them and to keep them for their protective value. Salivary glands make quite a bit of saliva, 1.5 liters, that's almost 2 quarts. It contains a water-based mucus, proteins, and the additional digestive enzymes. Now, these are just the initial enzymes. They are not the full suite. But we do get some substantial chemical digestion of those easily freed up nutrients in the mouth. It's important to lubricate the food. If you think about food, even if it's in its condition of being cooked, of being prepared, we often add oil. We often break down the watery substance, and the breakdown of the food material produces a sort of a slick material. But we're going to have to constantly lubricate that to keep it moving, because if it's dry, if it's unlubricated, it will stick. Dissolved food substances provide the taste, and taste has become an element of our enjoyment of life rather than our primary protective mechanism now. But all of that's going on in the mouth. Taste buds are an interesting consideration because the tongue has contradictory functions. It's handling the abrasive material and it's pushing it around. It's pushing it around and rubbing that material and moving it, meaning that the most wear that's encountered in the digestive tract is right there on the surface of the tongue before all that lubrication occurs. Now, to look at it in a little different way, the sense that we call taste requires a very delicate membrane and the binding of chemoreceptors. So it's not likely we're going to stick those chemoreceptors straight up on the surface of the tongue. And in fact, we don't. What we do is produce these bumps called papilla and these depressions. This is called the moat of the taste bud. We're going to take the sensory cells shown here, and we're going to line the vertical surfaces of the moat. You can see it here. Here's the moat, and here is the taste bud. This is a chemoreceptor, and these exposed membrane binding materials will bind to one and only one kind of chemical. So when we say it's sweet, sour, bitter, what do we say? Sweet, sour, bitter, meaty. There's one I've left out. I'll get it in a moment. That's because the specific receptors in that region only respond to that kind of chemical in the food. It is the dissolved material that is producing our sense of taste. That's why sweet and salt and acid. Acid is sour and, bitter is base, and then umami is more complex, but it is the dissolved material from meaty substances. Salivary glands. Oop, I went the wrong way, didn't I? The salivary glands are shown here. They're massive. The parotid right in front of the ear, the submandibular below the jaw, and the sublingual pump out large quantities, especially during eating. Now, saliva is an important sense of our part of our health. Our mouth should be fairly lubricated and moist at all times. Dry mouth is something that changes the conditions of the mouth and makes you susceptible to everything from tooth decay to other types of infection. Salivary glands can also be infected. This is a case, something you probably have never seen. This is a child with the mumps. The salivary glands have been infected by the mumps virus, and they swell. The entire neck swells up. It aches. It's very sensitive to temperature. It's sensitive to touch. I know that when mumps occur, generally it's a kind of a debilitating disease. The child just feels very bad, not like moving around. But in general, in children, at that stage of the development of the lymphatic system, there is not a substantial danger. However, if you survive childhood without contracting the mumps or being vaccinated, and contract it as an older adult, it can move through the glands of the body below the base of the neck. And especially in males, in the testes, it can provide a disease episode that will sterilize the male. So in adults, it is a more dangerous disease. Teeth are a story in their own. It's why we have a medical practice called dentistry, which is specializing in teeth. And this just illustrates the variety, kind of the plasticity between different species and how teeth have adapted, because it turns out that teeth are the onboard tools of non-human animals. They are their main and often only kind of mechanical digestion. So snakes have no arms. See how the teeth point backwards? This lower jaw can move forward and pull, and then the upper jaw can move forward and pull, and those teeth basically are the hands that push the prey item into the throat. The dog has shearing teeth, is a meat eater, and killing teeth. Here, the canines, named after the dog, that reflect its diet. Deer are herbivores, so here are the snippers, and then we move the grass. Grass has a lot of nutrition, but that grass must, absolutely must, be ground fine. You have to break enough cell walls that you can squash the membrane cell inside and release the nutrients. So big flat molars in long rows and a large jaw is what you see in deer and in cows and other grazers. The beaver uses its front teeth better than an axe. It basically can chew through a tree trunk. If you keep the beaver in a zoo, you have to give it wood. You have to place wood for it to chew, or its teeth basically will grow to the point where it injures itself. The elephant's canines have reversed and come outside as what we call tusks. These are used in everything from sexual display and contest to digging holes in dry riverbeds to reveal a surface of water. Finally, humans. Because of social, cultural factors, our teeth and jaws have been much reduced. Our jaw is shortened so that we have a flat face at maturity. I would point out that our close relatives like chimps and gorillas have that protruding mouth and jaw. It comes out here, all the way out here. And the reason for that is more jaw space, more chewing space, because they're eating raw leaves, raw plants all the time. We do so much food selection and food processing with our tools and softening with our cooking that these teeth are adequate, and I think that they have taken basically the selective pressure off of teeth and made them something that can move into a new realm. In the same way, our food, we care more now about the taste than the nutritive value. We know we'll get enough calories. And with our teeth, often we care more about the appearance of the teeth than we do about their actual function. We have two sets of teeth determined by genetics. Another animal that has the same thing is elephants. So that these small teeth, the deciduous teeth, will develop as the jaw, mandible, and maxilla as they grow. And the replacement, the size of the teeth, will match the size of the jaw, and the emergence, notice when these come in, will match the development of the jaw. They come in when there's enough room. Now, as the jaw continues to grow to this larger and larger structure, we'll replace these teeth on a kind of an alternating schedule, forward and back, with the third molars and wisdom teeth being the last ones as the jaw elongates. This bicuspid organization is one that provides us with our permanent set of teeth. And in fact, we now have good enough dental care and hygiene, and the fluoridation of water, all of those contribute to very long-lasting dental health, an important element of our health. We also have prosthetic substitutes so that we can produce teeth that are artificial teeth but function well in our feeding. The tooth itself is a bony structure. Dentin is more like bone. Enamel is a special exudate that hardens and protects the outside of the tooth. I would point out, enamel is the hardest substance in the body, harder even than bone. The tooth is secured into a socket with a cement layer attached into strong collagen fibers, the so-called periodontal ligament. Notice the kind of two-root structure that increases the surface area by having two roots, and also by allowing them to descend deep into the bony socket, the so-called alveolar process of the mandible or maxilla. We're going to provide the material for good tooth health through the delivery of resources in the blood, arteries in and veins out, and we're also going to make the teeth sensitive by the presence of nerves within this so-called pulp cavity, where the living cells are polymerizing both of these materials. Seal it with a fleshy growth, the gingiva or gums, and the tooth is complete. It is these gaps, it is the contact points between the teeth where food is lodged, and if not cleaned out, attracts increasing levels of bacteria. Those bacteria can damage the tooth by producing dental caries. Here is a micrograph of the bacteria and an extended, or not extensive, but advanced state of bacterial growth. In fact, the bacteria you notice here are diverse in their shape and their identity, and they produce an actual layer called dental plaque. Now, a clean tooth surface will only host a certain number of bacteria. If you allow this plaque or these biofilms to develop, it produces a new environment, including anaerobic environments, and new bacteria are recruited just by breathing and eating. These bacteria can have a bad effect on the teeth and cause chemical reactions like the production of tartar and calculus, which are hardened plaques and definitely indicate an advanced state of bacterial growth. Tooth decay will progress, showing erosion of the surfaces, showing splits developing within the enamel. Infected gums, as shown here, will begin to recede and produce these pockets. This is a very badly affected and infected mouth. Periodontal disease can lead to the loss of teeth, receding gums, and other problems. Prevention. It's very interesting because when we live our life in a certain way, we kind of figure humans have always done that. Well, it turns out they have not. We have not had refined sugar in such high amounts throughout the history of the human being and its populations. What we see is that when we start to introduce unlimited amount of sugar, and we start to feed on that sugar in unrestrained ways, that the amount of tooth decay, as shown by examining the teeth in grave goods, the amount of tooth decay goes way up. It's basically, a sugary treat is like painting syrup on your teeth, which is a perfect medium for bacterial growth. You think about your mouth, most of the time, it is dark, it is moist, and it is warm. Add sugar, and it's a perfect place for bacteria to grow. If you, however, take a sugary snack and clean up immediately, clean the surfaces and clean the gaps, that can control this factor. Floss is underrated in that the contact points, and especially the junctions between the gums and the teeth, are especially susceptible to decay. And so floss actually improves your prospect of good dental health in a large degree. Now, we've been at this about an hour. So what I'm going to do is I'm going to end the first part of the show and take a short break. And we will return